my objectives, methodologies I have used, main findings, and conclusion with some references. So without further ado, I'm beginning with the introduction. So bioactivity through computation. So for that, we must know what is bioactivity. Bioactivity means special type of ability to which is exhibited by a chemical compound on living organism, which could be harmful, beneficial, or neutral depending on whatever the uses it might have. For example, I have stated aspirin because it is widely used as analgesic, antipyretic, and anti-inflammatory properties. I have chosen the bioactive compounds. We have given the task to choose some compounds. So I have basically chosen citrulline, Sakuranetin, and Benzylisothiocyanate. So citrulline is found in watermelon in form of citrulline malate, that is citrulline and a combination of malic acid. It helps us in vasodilatory properties, reducing fatigue, our cardiovascular uh, functions, and the male reproductive health. Sakuranetin acts as a phytoalexin and it is used by rice plants and cherry blossom plants as a defense mechanism. Its use is the prevention for neurodegenerative diseases and also in improving our lipid metabolism and reducing the risk for cardiovascular diseases. Benzyl isothiocyanate is a naturally occurring constituent for cruciferous vegetables, and since it is a food additive, we can say that it enhances the shelf life of food products and also decreases the growth of foodborne pathogens. Now, the objectives of the entire project will be to use the ChemDraw software and the registration of Pass Online website, which help us to predict the bioactivities for chosen compounds. Which is for citrulline, I have chosen these five, namely protein disulfide reductase inhibitor, arginine 2 monooxygenase inhibitor, OMTIN inhibitor, yeast ribonuclease inhibitor, mucocystis treatment. For Sakuranetin, I have used CYP1A1 inhibitor, the substrate of its isoform, UGT1A10 substrate, and HMOX1 expression enhancer. For benzyl isothiocyanate, I have used alkene 1 monooxygenase inhibitor, chemoprotective agent, anti-neoplastic, and anti-neoplastic for lymphocytic leukemia, and how it is used for phobic disorder treatments. Next, for the methodologies which I have used, I have first, this is the ChemDraw software, which is used for designing the compounds. The registration for Pass Online website, which is currently I am logged in. Secondly, the Marvin J's panel for citrulline, for benzyl isothiocyanate, for Sakuranetin, and the bioactivities which are listed below. I have selected 5 among them, and today I am going to present the main findings on 3 of each of the compounds. So the main findings for citrulline, which I am going to present about, will be arginine 2 monooxygenase inhibitor, OMTIN inhibitor, and how it is an important use for mucocystis treatment. Arginine 2 monooxygenase inhibitor is the main inhibitor in which the L-arginine, which is the pre-reactant for citrulline, recouples with the nitric oxide synthetase, which increases the nitric oxide production. Basically, these inhibitors help in increasing and just modulate the levels of citrulline within cell for various biological purposes. The OMTIN inhibitor is not related directly to human physiology, but physiological processes of bacteria in which it actually inhibits the virulence mechanisms and enhances the host immune response, since bacteria live inside our body, and the omptin inhibitor, it protects our body from bacterial infections and pathogenesis. Mucocystis treatment is a real life phenomenon in which mucocystis is considered as the inflammation of the line in our mouth and the gastrointestinal tract. Oral mucocystis might be mild or severe, but the gastrointestinal mucocystis could be manifesting abdominal pain and nausea. So citrulline comes in action, promising non-invasive biomarker. Non-invasive biomarkers means those biomarkers which can be used from our bodily fluids and tissues without excluding the operational part, which is very necessary for cancer treatment. The next one will be Sakuranetin. Sakuranetin is found in cherry blossom trees, rice plants, etc. The main findings which I am going to explain are CYP1A1 inhibitor and CYP1A1 substrate, HMOX1 expression enhancer. So the CYP1A1 inhibitor is actually cytochrome P450. The 1A1 is the most important for detoxification enzymes in our body. Inhibitor means it only catalyzes our detoxification mechanisms. The substrate part can catalyze the hydroxylation of the flavonoids, that is the position of the molecules. And HMOX expression enhancer is basically hemioxygenase 1. That enzyme plays a critical role in hemicatabolism, and which is converting the heme into biliverdin, releasing carbon monoxide and free ions. It is a cytoprotective enzyme with antioxidant, anti-inflammatory, and anti-apoptotic properties. Nextly will be benzylisothiocyanate. The main findings which I have listed here will be chemoprotective agent, anti-neoplastic, and phobic disorder treatments. Chemoprotective meaning how can we prevent the side effects of chemotherapy. BITC, in short for benzylisothiocyanate, helps us in vitro molecular mechanisms, and it potentially decreases the permanent damage that chemotherapy has caused into our bodily tissues. Antineoplastic is the further treatment in which we inhibit the prevention and growth and the spread of the cancer cells, that is the abnormal growth of tissues, and which we referred as cysts and tumors. These antineoplastic agents try to kill directly the cancer cells and target the specified pathways which can in future crucial for cancer growth and survival. Phobic disorder treatment, on the other hand, will be an overwhelming application for benzyl isothiocyanate, because in modern generation phobia has become a serious issue. BITC can mitigate our neurobiological mechanism and the underlying phobic disorders, and also the problem with anxiety as well. So in conclusion, I will conclude that the fusion of computational techniques with the study of bioactivity is a revolutionary approach, and through this project, I tried to emphasize what I could in this short period of time, and the compounds which I have chosen have a vast area of research, and for future purposes, this will be a very beneficial stepping stone. The references which I have used are listed down here, but mostly, to be honest, I have used AI language to just to create sentences and definition for the basic explanation which I am giving now. Now I am ending my presentation with the gratefulness to my teachers, Miss Nivedita ma'am, Miss Snigdha ma'am, and my senior